So we'll continue with statistical analysis, where in this case now, we want to compare between two sets of data. So when we do experiment, there are different ways to get the data. So here, start means when you start to do experiment, it could be your data is in the form of frequency. Frequency means number of individuals. Okay, number of individuals. For example, in your class, how many students, how many number of students is a blood group A? How many number of students is blood group B, A, B, and O? So it's number of students, right? So the frequency. So you're talking about frequency of, for example, in this particular hill, right? For this plant, what is the population size of this plant in the bottom, in the middle, and the top of the hill? So it's dealing with number of organism, number of individual. So because of that, the data, right, is actually discrete or discontinuous, because it's individual. It must be 1, 2, 3, 4, 5. It cannot be 2.34. So therefore, it's a discrete number, no decimal one, and it's discontinuous. And then the data is also categoric. That's my category. Now, one more thing. Okay. So the data is discrete or discontinuous, and it's also categoric because I'm talking about blood group. So there are A, B, and O is called the category. Or in the hill, bottom, middle, top of the hill is also categoric. So the data has got this nature. Number one is discrete. Number two is categoric also. So if my data is in this form, and then I've got an expected outcome for my data. Expected outcome means to say that, okay, in my genetic, so let's say my experiment is genetic crosses experiment. So I have got the expected ratio, right? Which I calculate using genetic diagram. Let's say expected ratio is 1 to 1. Then I go and conduct experiment in the lab. I cross the fruit flies and I count the offsprings. So I got an observed ratio. The offsprings have got, let's say, 13 normal wings and then 24 vestigial wings, say for example. So that will be by observed data. So there is an expected outcome, which is the E. So I want to know whether the observed data is significantly different from expected data or not. What test should I do? My dear, what test should we do? Chi-square test, right? So we have to do chi-square test. So for this one that I want to find out, I must do a chi-square test. So I must do chi-square test. Okay. So this is for experiment, which is a genetic cross. Of course, second experiment is also ecological sampling. For example, this herbicide, I want to know the effect of introducing this herbicide in this field to this particular plant population. So therefore, when the time is 0, at time 0, time equals to 0, when the herbicide has not been introduced yet, what is my sample size for this plant? Maybe I have got maybe 44, this type of plant in this area. After it has been introduced for 2 years, I find that my total number of plants becomes 30. And then after introduced for 4 years, the total number of plants becomes, let's say, 10. So I got 3 readings here, right? So how do I do my chi-square test? Because I can see my sample, my data is discrete number, right? And also my data is categoric because it's divided into time equals to 0, time equals to 2, time equals to 4. It's categoric. So therefore, in order to see whether the introduction of herbicide significantly caused the decrease in the plant number, I must do chi-square test. So my question here is, for this chi-square test, what is my expected ratio? because I already got my observed ratio is equals to 44 to 30 to 10, correct? My expected ratio, sorry, my observed ratio is 44, 30, and 10. So can you tell me what is the expected ratio that I should put for this chi-square test? So my assumption is it will be usually for ecological sampling. Your expected ratio will always be 1 to 1 to 1. So when you do chi-square test for ecological sampling, then usually is 1 to 1 to 1. Why? Because my assumption is the introduction of the herbicide does not affect the population size of this plant. So this is what I want to test. Then after that, I get the observed ratio. I do my chi-square test. After I do my chi-square test, if I found out that O is significantly, so sorry, there's 95% confidence. There's 95% confidence. This is my conclusion. That O is significantly different from E. I'm not going to write. I'm just going to use the formula. So you got 95% confidence that O is significantly different from E. What is your conclusion? Does the herbicide significantly reduce or does not reduce the population size of the plant? Hello? Who said significantly reduce? Raise up your hand. Who said does not significantly reduce? Raise up your hand. Okay. Now, you know that chi-square test is a goodness of fit test, right? Goodness of fit means you want to know whether your O equals to E, correct? So once you've done your chi-square test, your conclusion is you got 95% confidence that O is significantly different from E. So can I say my 44, 30, and 10 is equals to 1 to 1 to 1? No, right? It cannot be 1 to 1 to 1. That means to say, is there a difference already? Yes. So that means to say, is the herbicide giving these differences? Yes, isn't it? Because if the herbicide does not cause a difference, we will expect the ratio to be 1 to 1 to 1 already. That means O must be equals to E. But now we found out from chi-square test, O is significantly different from E. That means I cannot take this ratio here as 1 to 1 to 1 already. There's a difference already. So the herbicide actually affects the plant population size through time. As the time passes by, the total number of plants actually drops. So the herbicide actually causes the decrease in the population size of the plant through time. So that's the conclusion we can make from the ecological sampling for chi-square test. So usually chi-square test can be used to do for genetic process experiments or ecological sampling experiments. These two. Okay. So this is for chi-square test. So other than this kind of experiment, sometimes the more familiar experiment for you is in the lab, you do measurements. For example, what is the distance traveled by manometer fluid, right, in 1 minute, for example? What is the total number of bubbles released in 5 minutes? Okay, what is the time taken to reach endpoint? Okay, so these are all measurements. So the experiment involves taking measurements of a particular variable. So the data that you obtain is quantitative and it is continuous. Continuous, for example, distance traveled by manometer fluid. It can be 12.3, and then after that, 12.3 cm. 12.3 cm. Okay. Okay. And I said the one 12.3 lah. 12.3 mm. 12.3 mm. So it can be for 15.9 mm. So it's a continuous value one when you plot on the graph. It's not discrete. And then the data, they are not categoric. There's no category. It's only number of bubbles released, distance traveled, full stop. So there's no category there. Okay. So let's say, for example, 2 students make the measurement. 2 students make the measurement. Say at 20 degrees Celsius. 20 too small. Let's say 40. 30 degrees Celsius. So at 30 degrees Celsius, we run the respirometer. Okay, so two students set up the same thing, run it for 5 minutes. Okay, so student A, and then this is student A. Okay, student A collect the data, which is the distance travel. So let's say distance travel is 12, and then this is repeat experiment, okay? 14, repeat experiment again, 9. So there are 3 readings taken by student A, and then student B also take 3 readings. She got, let's say, 17, 24, 9. Okay, let's say for example. So therefore, this is a replicate, right? Reading number 1, reading number 2, reading number 3. So therefore, they will calculate the mean value for their reading. So I will have mean value from student A and mean value from student B, right? So after the mean value is obtained, I want to see whether there's a significant difference between the result from these 2 students or not. Okay, whether there's a significant result from these 2 students. Sorry, I want to change it. Let's say this student conduct experiment at 40 degrees Celsius, I think. So I want to know by increasing the temperature, will it actually increase the rate of respiration? So I've got 2 data here, and then therefore I got 2 mean. So this is the mean at 30 degrees Celsius, and this is the mean at 40 degrees Celsius. Okay, so this is the mean. So I want to know whether there is a significant difference between these 2 mean, because based on theory, every time when temperature increases by 10 degrees Celsius, the rate will double, right? So I want to verify whether it's really true from my experiment. So how do I compare whether these 2 sets of mean are significantly different or not, so that I can conclude that when I increase temperature from 30 to 40 degrees Celsius, it actually increases the rate of reaction. So I have to do a test, right? So the test that we're going to learn today here is actually called the t-test. Use t-test. Okay, so now we go to page 3 of your notes. What is the use of t-test? So t-test is used to compare 2 sets of samples if they are significantly different or not by comparing their means. By comparing their means, and we also need their standard deviation value. So for example, when do we use t-test? So when we have 2 samples that have been measured in the same method like just now, measurement is the same except for temperature has changed. So we want to find out whether the mean of A is equals to mean of B. For example, the grass density on the top versus the bottom of the hill, the white blood cell count when a person is infected versus an uninfected human, the fetal weight in a smoking mother versus a non-smoking mother, whether it's a difference. So you can find that the data is a measurement that's continuous, meaning not discrete. And then the data must be normally distributed. Normally distributed means this mean value is calculated from a set of data, right? So the set of data must be normally distributed around the mean. Okay. So this is mean of A lah. So the rest of data are distributed normally around the mean of A. Okay. So t-test will only be used when the total sample size is between 3 to 30. That means you minimum must have 3 readings, maximum 30. So this is for t-test. So let's see how to do t-test. If you know chi-square test, t-test is almost similar. The steps involved they are quite similar. So step number 1, let null hypothesis be HO be mean of A is equals to mean of B, meaning to say that there is no significant difference between mean of group A and mean of group B. So after that, calculate standard deviation of the sample data. So use this formula to calculate. Now what I want you to do, I want you to change the n here, change it to I. This n is supposed to be I, so change it to I. Okay. Do you know the meaning of this formula or not? After I show you, okay? So calculate standard deviation. So you already know that standard deviation is the distribution of the data around the mean. So how far the sample is distributed away from the mean. So next step is after calculate standard deviation. Okay, so this is an example that I want you to do now. Please calculate the mean value of this set of measurements for sample A and this set of measurements for sample B. Calculate the mean. After that, calculate the standard deviation. I will also show the working for this one. Standard deviation is this one, this working, okay? Just in case you are not quite sure. So the mean is 0. Start from here. So this is the mean. Minus the first reading is 0.76. Square it, square it, then plus 0.736 minus second reading 0.74. 0.74. Square it plus 0.736 minus the third reading is 0.75. 0.75. Square it plus 0.736 minus the fourth reading is 0.72. Square it plus last one 0.736 minus the last reading is 0.7. Square it. So this is for this part here, summation of the mean minus xi is the number of reading, the first reading, second reading. So minus the first reading, mean minus the second reading, mean minus the third reading, mean minus the fourth reading, mean minus the fifth reading, square all of them. So summation of. Okay, you can get it up. Then after that, for standard deviation, you have to root square and then divided by n minus 1. So after you have done your calculation for standard deviation, the next thing is you can calculate the t-value already. So find out the sample t-value. The formula will be given to you. So what you need to calculate t-value is, of course, the mean value and the standard deviation value and the sample size. So can you calculate now and see whether you can get 3.76? So we have calculated t-value is 3.76. What's next? Similar to chi-square test, now we want to locate the location of this t-value in the t-value graph, whether it's in the rejection zone or acceptable zone. So let's put it into the graph. So this is a t-distribution graph, right? So this is right in the center where the t-value is equal to 0. So let's recap again. Okay, so when t is equals to 0, based on the formula, that means A, mean of A must be equals to mean of B, right? Based on the formula. Look at the formula. T is equals to this formula. So when t equals to 0, it's because A is equals to B. So this is 0. So t is 0. So right in the center of the graph, when t equals 0, that means mean of A equals to mean of B. So probability of mean of A equals to mean of B is actually exactly 100%. So as the t value starts to increase, so t values start to increase, that means to say the differences between mean of A and B will start to increase as well. So that means the probability of mean of A equals to mean of B will start to drop. So we know that those data at the end of the graph are the extreme data that's very far from the mean. So these two are the extreme data that are very far from the mean. It can be higher than the mean or lower than the mean. So therefore, where are the locations where we're supposed to reject the data because they're too far from the mean? So it may not be accurate anymore. So we reject the 5% n, isn't it? So because the graph is on both right and left side, that means the right side is 2.5% and the left side is 2.5% n. Okay? So this first thing I want to let you know. So the zone of rejection is the shaded in blue color. So every time you get your t value over here, your conclusion is there is a significant difference between mean of A and mean of B. Whereas over here, there's no significant difference between mean of A and mean of B, because this is what you're testing, right? Okay. Now, another thing I want to bring to attention is when we calculate the t value, do you see that we have placed these two bars across our calculation? That means we have already converted all the negative value to positive value, right? So therefore, because of that, we have to adjust the graph just now. So we must bring all the negative value on the left side of the graph to be included in the positive region. Okay, so all this negative, we have to bring it to the positive side. That means this part here will now be 5%. And this part here will now be 95%. And we no longer have this part. So this part don't have reading. Okay, based on the calculation. So therefore, you look at the t table. T table are all positive value because of this reason. So now, how to do conclusion? When the sample t value of the sample is equals to or higher than critical t value. So this is critical t value, which is a cutoff point. When the property of mean of A equals to mean of B is exactly 5%. So this is a cutoff point. So when a sample t value is equals to or higher than critical t value, that means located here, probability of A equals to B will be less than or equal to 0.05. So you reject now hypothesis. So you have 95% confidence that mean of A is not equals to mean of B. So this is the full conclusion that you can do. But when a sample t is less than critical t, that means sample t is located in this region here. So therefore, that means probability of mean of A equals to mean of B is more than 0.05. So we accept null hypothesis. So we have 95% confidence that mean of A is no significant difference from mean of B. So very similar to chi-square test. So if you're familiar with chi-square test, t-test is actually very easy. So let's do the sample that we have. 3.76, right, is the t value. Okay, next step is, after you calculate your t value, find out the degrees of freedom. So the degrees of freedom here is, because we got two samples, each of them also got their own degrees of freedom. For sample A, degrees of freedom is n minus 1. Sample B is n of B minus 1. So the degrees of freedom is the addition of both their degrees of freedom. So therefore, it's NA plus NB minus 2. So this is the only difference between chi-square test and t-test is the degrees of freedom formula is different. In chi-square test, the degrees of freedom is equals to C minus 1, where C is number of category. So we've got 5. Total number of samples is 5. So 5 minus 1 plus 5 minus 1 is 8. So the next step is find out the probability of mean of A equals to mean of B in the T table. So where is the location of my T value in the T table? Is it in the rejection zone or acceptable zone? Rejection zone means reject null hypothesis. Acceptable zone means accept null hypothesis. Okay, so my T value is 3.76. Degrees of freedom is 8. Let's go to the T table and find its location. 3.76 and 8. So there's a T table here. So this is a T table. So the body of the T table shows the T table value. I'm at degrees of freedom 8. So I must look at degrees of freedom 8 is here. This degree of freedom 8. And my T value is 3.76. So 3.76 is located here. Right. 3.76. So let me put here, this is my value, 3.76. So it's here. So conclusion. What is the probability of mean of A is equals to mean of B is between these two, right? So after I found out the probability, I can do the conclusion already. So the probability here is between 0.001 and 0.01. So let's look at how we write the answer, okay? Okay, so now from the graph, I have already found out that the probability is between 0.01 to 0.001 to 0.01. So meaning to say that probability will be less than 0.05. So this is the first way when the table is given. T table given. Then this is how you do it. Second way is critical T value given. Then they don't give you the table, they give you the critical T value only. For example, critical T value is actually 2.31. 2.31. So I'll show you how to see 2.31, whether it's correct or not. So degrees of freedom 8. Critical T value is when probability is at 0.05. Degrees of freedom 8. So it's therefore this value here. It's 2.31. So this critical T value. Okay. So critical T value is 2.31. My sample T value that I've calculated is 3.76. So my sample T value is higher than critical T value. Higher means it's in the rejection zone, right? Higher means the probability is less than 0.05. Probability of what? Probability of mean of A equals to mean of B. So how to do conclusion? Whenever the probability is less than or equals to 0.05, we will reject null hypothesis, meaning to say there is a significant difference between mean of A and mean of B. Whenever P is more than 0.05, we will accept null hypothesis, meaning to say that there's no significant difference between mean of A and mean of B. Since the probability is more than or less than, sorry, since just now, this is like just now, since the probability is between 0.01 to 0.001 to 0.01, so therefore the probability is actually less than 0.05. So when it's less, we will reject the null hypothesis. If you actually write what is null hypothesis, then you say reject null hypothesis. If you don't write reject. Okay? Because if you don't write null hypothesis, if you say reject null hypothesis, I will not know what you mean by reject null hypothesis. You must have a null hypothesis statement first. Then when you do conclusion, you can say you reject. Okay. So reject means what? Reject means there is 95% confidence that there is a significant difference between the mean of A and mean of B. Okay.